right. Wonderful. Cool. That should be recorded. All right. There we got you.
All right. Awesome.
Let me see if I can make this full screen.
Okay. Let's see. This is different than what we tested. This is like the Hangout Air thing instead of a. So let's see if we can figure out how to do screen sharing from here.
If you go to the left,
Yeah,
there's a little green square will pop up.
Yeah, I got it.
I want to make all this stuff go away.
Let's see.
So we still have all the stuff.
Still all the stuff. You present. Cool. How's that look?
That's better. All right, Roo, we should be good to go.
Awesome. Sweet. Can you guys hear me okay?
Yeah.
Yeah? Excellent. Well, I can't see you at all, so hi, everybody. Well, my name's Roo Vandergrift. I'm a recent PhD graduate from University of Oregon. I studied Xylaria for my PhD. So I'm gonna kind of go through. So we're probably mostly familiar with Xylaria hypoxylon as our most common North American species. Out there on the East Coast, you guys are a little bit lucky in terms of Xylaria compared to the West Coast here. With a more depauperate flora in the West, more interesting species on the East Coast. I kind of want to talk about sort of the what, the where, and the how for Xylaria. What are they? How do you recognize them? Where do they occur? Talk some about diversity in Xylaria, and then talk about how you identify them, what they're good for. Okay. Would somebody maybe just for unless there's a question, could somebody mute the mic on that end? I'm getting some feedback through it. Awesome, thank you. So let's start out just kind of briefly thinking about classification. What is Xylaria? And we know fungi we can split broadly into basidiomycetes, ascomycetes. Basidiomycetes have basidia. Ascomycetes have asci, which are the spores. So Xylaria are ascomycetes. So in that first broad split, Xylaria are ascomycetes. Spores are born in asci. They tend to exist in haploid state, no plant connections to the mycelium. And the parathecial ascomycetes. So within ascomycetes, right, you split ascomycetes broadly into the classes of those with apothecia and those with parathecia. And so classification-wise, Xylaria, the spores are born in parathecia embedded in a stromata. So each ascomata is a parathecium. So these are Sordarioomycetes, and they're actually the type for their own order, the Xylariales, which includes a bunch of families you've probably never heard of, including Amphispherieliaceae and things along those lines. So interesting critters. Parathecial stromatic ascomycetes. And there's some other things that are parathecial ascomycetes that you might be familiar with, right? Hypomyces, the lobster mushroom fungus. If you get real close on a lobster mushroom, right, you'll find the little parathecia on the surface. There are other parathecial fungi that occur all over, including things. This is a Xylaria that's being parasitized, much like a Hypomyces, by a different parathecial fungus. This is in the hypocreales. This is a Nectriaceae that parasitizes these xylariaceous fungi. Right, so how do we recognize xylarias? So Xylaria is a stromatic ascomycetes with embedded parathecia, and they're characterized by having carbonaceous is the kind of technical term. Carbonaceous outer coating. There often is also a carbonaceous coating around the parathecium, and then they have single-celled ascospores. Right, so when you look in the microscope, you'll see the cells are single-celled. They're what's called ellipsoid inequilateral, which just means flattened football shaped, right? And they're dark. The spores are heavily melanized, and they're relatively large. When we think of, I don't know, probably if you look at a basidiomycete, you're looking at spores 10 micrometers long. A lot of xylarias have spores 30 and 40 micrometers long. Spores can be relatively large, and they have a germination pore or slit, usually a slit. And this kind of clear line in these spores, and that's where the spore germinates from. And the germination slit, and then in the tip of the ascus, there's this ascus apical apparatus or ascus apex plug that is iodine reactive. It turns blue in an iodine solution, and you can see it. And these features, the germ slit, the size and shape of the ascospore, and the apical plug, are what we call taxonomically stable, which means that within a given species, they're the same from individual to individual. Whereas a lot of the macromorphology for Xylaria, there's some consistency within species, but they're hugely variable. They have just incredible variation in macromorphology, which makes them really difficult to identify. So you pretty much need microscopic features to be able to confidently pin a species on a Xylaria most of the time, which is a barrier for a lot of sort of amateurs interested in these fungi. And so let's look for a minute, like, what are they good for? So this is a tropical species from my field work in Ecuador. This is Xylaria schweinitzii. This is the closest tropical relative to Xylaria polymorpha. But the Xylaria polymorpha is the common hardwood species called dead man's fingers you guys see on the East Coast and the Midwest. And when they're young, they exude some exudates. They have an asexual conidial layer on the surface. And so most Xylarias have this development where they start out with an asexual layer on the surface, asexually produced spores, of which the function is, we can talk a little bit more about that afterwards. The function of those asexually produced spores is an area of active research right now. Nobody's quite sure what they're good for. And then as they develop, you get the parathecia. And then a lot of people, people ask me like, oh, well, can you eat them? And generally I say no, but lately there's been a few people that have started collecting Xylaria polymorpha, in particular on the east coast of the US, and using the young fruiting bodies mostly for flavor. The flavor is said to be very umami. There's particularly one person, Alan Burgo, apparently really enjoys it with steak tartare, which I don't know, I've been a vegetarian for a long time, so this seems like a little extreme to me, but so you can eat Xylaria polymorpha apparently. It's not traditionally eaten, and there's not much, it doesn't seem to cause intense gastronomic distress or any acute symptoms for people. However, Xylarias, this lifestyle where they have these long-lived fruiting bodies in places where generally fungi are relatively ephemeral, means that they need some way to protect those fruiting bodies from being immediately consumed. And so Xylarias are actually packed full of all kinds of interesting secondary chemicals. So they produce lots of defensive chemicals, xylenes and xylols, among other things, that are very unique chemistry to these fungi that are under a lot of investigation for use as drugs. There's some new cancer therapeutics that are based on chemicals isolated from Xylarias that are coming down the pipeline in Germany. And they're I mean, just hundreds of, my friend Mark Stadler, who's a professor in Germany, works on secondary chemistry of these fungi. And it's just, I mean, it's staggering, hundreds of interesting, unique, like very biologically active chemicals in these fungi. So I don't generally recommend eating them, just because there is lots of active chemistry that we don't understand. And it's not a traditional food source. This kind of like emergence of Xylaria polymorpha as an interesting food is not something that there is a history of. It's something that's developed really recently, and so we don't have, you know, the thousands of years of cultural understanding of the effects of this fungus the way we do with eating something like lobster mushrooms or chanterelles, you know, or even eating something like Amanita muscaria. Amanita muscaria is, you know, it's often listed as poisonous, but really it's just toxic. It'll give you some gastric upset and it'll make you trip. But if you boil it, all those chemicals boil away, and then you can eat it safely. It's a prized food source in Italy, and so there's you know, hundreds of years, thousands of years of tradition for how do you safely eat this food that we don't have for Xylaria. There are some species, Xylaria schweinitzii, that I showed you earlier, is used in some Andean cultures as a remedy for childhood bedwetting. And in China, Xylaria nigripes is used as a kind of a health tonic. They make tea out of it, and it has been used for a relatively long time. And that's from a very different taxonomic cluster, and we'll talk for a minute about that. The other reason Xylaria are kind of interesting, we're talking about like why do we care about Xylarias? You know, they're these crusty black things that grow on wood. And like, you know, so we can eat them. That's one reason we might care. I don't know if you'd want to eat them in any quantity that would be calorically meaningful. But we also, just the fact that they grow on wood is something we care about. That's something I care about. These, in tropical systems, Xylarias are one of the most abundant decay fungi for woody substrates. Some estimates put it at, they're responsible for 20 to 40% of decay of lignicolous, which just means like having lignicolous substrates in tropical systems, which makes them just huge players in the global carbon cycle, right? So if we're thinking about global climate change and global climate cycling, then Xylarias end up being this huge player that we don't really understand right now. And as part of that, they have this really interesting ecology. In tropical systems, they're almost ubiquitous endophytes, which is what I studied for my dissertation. An endophyte is just a fungus. It's a fungus that lives within a living plant tissue without causing disease. And so in tropical systems, if you take down any leaf from a forest like this and culture fungi out of that leaf, you're almost guaranteed to find a Xylaria eventually. They're ubiquitous common endophytes. Sometimes they're the dominant endophyte in the system, the most common one. And even we find Xylarias as endophytes even up here in the temperate zone. We did an endophyte study here in Oregon with Linnea borealis, this little, it's in the Caprifoliaceae, a little honeysuckle family ground vine in the forests in the Cascades. And we found several isolates of this Xylaria that at the time, nobody knew what it was. It's a species described in 2012 from Europe, Xylaria carsticola. And we find it as an endophyte here in Oregon. It's never been reported to fruit in Oregon. It's really interesting ecologically. And then there are other really interesting ecological associations. This is in that group with Xylaria nigripes, the one that's used medicinally in China. This is, I'm not sure what species this is. This is a photo from my friend Liz in South Africa. This is a group of soft-bodied Xylarias that fruit specifically on old termite mounds. It's a phylogenetically cohesive group. They're all relatively soft, and they have this really interesting ecological association with fungus farming termites. old world termites farm termitomyces fungus for the wood decay enzymes that they produce. And whether or not these xylarias that associate with these termite mounds are another fungus that the termites are farming, whether they're a parasite on the termitomyces, or whether they're kind of a sneakily utilizing the same substrate, is still unclear. There's a lot of work going on these fungi. They're really interesting. And then the other reason we're kind of interested in xylarias is because of this. It's this huge reservoir of diversity that we don't really understand right now. There are, at the moment, if you go into Microbank or Index Fungorum, which are kind of the two clearinghouses for names of fungi, for where they're places where all the names are listed officially, you will find there are more than 800 named species of Xylaria. A single genus, more than 800 species, which is one of the largest genera of ascomycetes out there, one of the largest genera of fungi out there. They diverged, they start diverging around the same time that the angiosperms start diverging, so it's a very ancient lineage, and there's just this huge diversity. The genus has never been monographed. There is no book, the genus Xylaria. There's no way, there's no single place to go look up what are these fungi. And we still, they've been relatively poorly studied. And so these two images that I'm showing you, this here is a Xylaria fruiting from sclerotia, and you see these large tuberous sclerotia. These were buried in a very deeply decayed hemlock log near Mount Hood in Oregon. And this is an undescribed species. This is a brand new species from our backyard in Oregon. This here, this is a species of Xylaria that's been reported since about 2012 from the Northwest. There's a researcher up in Washington, Brenda Callan, has suggested the name Xylaria atropictor, but this species is yet to be described. There is at the moment no real species called Xylaria atropictor. There are a few people finding them and giving them that name because Brenda's put the name out there. But this is an active, this needs to be described. And so here we have two species of Xylaria, large macroscopic charismatic fungi from Oregon and Washington, a place where the world expert on Xylaria, Xylariaceous fungi for the whole family, is this person, Dr. Jack Rogers, who just retired last year, or no, 2 years ago, from University of Washington up in Pullman. And so these are two Xylarias from the backyard of the longest working expert on this family that's existed ever. And they're undescribed still. He never found them. So there's, one person can only do so much. And so one of the reasons where I'm interested in these fungi, and I think we should all be interested in these fungi, is because there's this incredible diversity out there, ecological habits, and just new things we can find. And who knows what good these things will do us. These have really, one of the things that's really interesting about Xylaria ecologically is that they're lignicolous, they decay lignin. And we think of most lignin decay fungi as being basidiomycetes, right? White rot fungi are generally basidiomycetes. They use this lignin peroxidase enzymatic pathway to break down lignin. Well, Xylarias are this ancient ascomycete lineage that decays lignin. They're white rot fungi, and they use some other mechanism that we don't understand. We don't know how they eat lignin. And that's it's a huge, if you can figure that out, that's a huge contribution to the literature. And those enzymatic processes, right? The enzymatic processes that allow fungi to eat lignin are the same things that allow fungi to eat plastic. And so Xylarias have the potential to be a really useful area of research towards bioremediation that's totally untapped at the moment. Oh, some more diversity. These are two species from my field site in Ecuador. This one on the right here, you can see is giant. It's a 6-inch ruler. And this one on the left, I don't know if you can tell from this little snip of substrate, this is growing on bamboo, which is a monocot, not a dicot, right? which is kind of a peculiar substrate for Xylarias. And so these two are, again, undescribed species. This is diversity that has not yet been documented. So I'm working on writing these up. Right? And Xylarias, I mean, they have this amazing diversity of form and function, from the very, very tiny. This is a great picture from Danny from our site in Ecuador. This is Xylaria tucumanensis here on the left. And you can really see the perithecia with the osteolar openings really clearly here. And this here on the right is Xylaria globosa, which is characterized by these red exudates. And so Xylarias have really interesting ecological associations as well with insects. In the New World, in tropical America generally, we find Xylarias in association with these flies, and we think that the red exudates for Xylaria globosa are a mechanism for attracting these flies. And whether or not the flies are serving as dispersal agents, or my personal theory that they're serving as pollinators, quote unquote, moving sexual propagules around the conidial surface, the little asexually produced spores. One of the theories for what they might be doing is serving as pollen. And so when the flies move them from fruiting body to fruiting body, they may be moving around gametes instead of propagules. But that's again active research. We don't know yet. We know they're associated with these insects. We know that they do things like produce these exudates, which are full of all kinds of interesting polysaccharides, sugars, but we don't really know why yet. There's a lot of unknowns in Xylaria. It's a mysterious group. And part of why, when we're thinking about that, when we're thinking about this classification, is it's the Xylaria, and the Xylariaceae in general, has been kind of a mess taxonomically. There aren't terribly many morphological characters to work with because the fruiting bodies are so morphologically variable. And so when we look at a phylogenetic tree for Xylariaceae, which is what we're looking at here, you see that it's split broadly into two big branches. On the top, we have what's called the subfamily Hypoxyloideae, of which Hypoxylon is the major group. And on the bottom, we have this large group, the subfamily Xylarioideae, of which Xylaria is the major group. And Xylaria, the thing that's confusing about Xylaria is paraphyletic. There are three major clades within the genus Xylaria, which wouldn't really be an issue, except that there are other genera that arise from within those clades. So some of you might be familiar with the genus Kretschmaria, which in North America is most often encountered as Kretschmaria deusta. Kretschmaria is like big flat Kretschmaria. So Kretschmarias are very closely related to Xylarias, and there are Xylarias, phylogenetically speaking, on either side. Same thing, some Rosalinias appear to really just be uniparitheciate Xylarias. We'll show you an illustration in a minute. And same thing, the genus NoMania, which for a long time was classified as a hypoxylon. NoManias, it turns out, are really just Xylarias that are laying flat instead of upright. And then the Xylarias themselves, there is this subgenus Pseudoxylaria. These are the termite-associated ones, the soft-bodied ones. And they're basal diverging within the Xylaria group, and very distinct phylogenetically. They're their own clade. So I suspect at some point in the near future, the subgenus Pseudoxylaria is going to be elevated to the status of just genus Pseudoxylaria. And then the Podosodaria and the Peronia are upright fruiting bodies that tend to occur on herbivore dung. You find these on deer or cow poop. So keep your eyes open. Horse, you find them in horse poop. So those are really interesting. And basal diverging. And so Kretschmaria deusta, in the Northwest, you find it on big leaf maple. On the East Coast, you'll find it on a few other things, but largely maples. And Kretschmaria deusta, often people see it when it's in its young phase. And again, it has, just like Xylarias, it has two different reproductive modes. It has an asexual spore stage on the surface, and that's what this gray with the white border is. And then as it matures, the conidial, those asexual spore layer, sloughs off, and the perithecia develop inside a carbonized outer coating underneath. And that's what this black is. This is the carbonized outer coating, and then there's little perithecia embedded. And so you often will see one form or the other. It's kind of rare to see it both together like this, at least on the West Coast, because of our strong seasonality of precipitation. Rosalinias are characterized by these single perithecia embedded in they call it a subiculum. There's a little fuzzy coating to the bottom. And the Rosalinia is there was a recent monograph of Rosalinia in 2013, I think. Liliane Petrini, the European researcher, is the world expert on Rosalinia. And she wrote this great book on Rosalinia. I think it's just called Rosalinia, a World Monograph. If you're interested in these little fungi, I highly recommend that book. One of the things that book really tells you is that these fungi, much like Xylaria, are very understudied, undercollected. And so she monographs 140 species of Rosalinia in the world, and about a third of them are represented by just a single collection, only known from the type, which is wild, right? Only known from the type specimen. I mean, and there's not really genetics. Rosalinias, because they're small, they're hard to culture. There's not really much in the way of genetics for Rosalinia yet. It's getting there. It's getting better as time goes on. But right now, all the classification for Rosalinia is morphological. And we know for all of fungi that morphological classifications are often incorrect because morphology in fungi is a little bit more variable than in animals or plants. There are sometimes less characters, and there's a lot of convergent evolution in the fungi. And so there's a big group of Rosalinias that we do have some genetics for that really appear to be just uniparothecia Xylarias, really reduced Xylarias. And the third group that was arising from within Xylaria are NoManias. NoManias, you can see why they were called hypoxylons for a long time. This is Nomania bipapillata. NoManias are really just aplanate Xylarias. That's another, but it is a cohesive group, and it's reduced. It's this reduced Xylarias where they're down to just a few perithecia sometimes, and they've lost their ability to be upright stromata. But when you look at the spore morphology, when you look at those ascus apical plugs, you find that they resemble Xylarias. And then when you generally NoManias, one of the ways you can tell NoManias from hypoxylons is that usually when you cut into a hypoxylon, the inside of the fruiting body is dark colored and they have waxy granules underneath the surface. NoManias don't have any waxy granules and they tend to be white on the inside, just like Xylarias, right? So Xylarias tend to have white interior stromatic tissue. And so this is that Xylaria globosa that I was showing you earlier with the red exudates. And so this is the fruiting body that we had that photo of earlier. And so the question then is, what ties Xylaria all together if there's all these different genera? And so Xylarias, right, just to remember, Xylarias 8 ascospores to an ascus, this apical plug that stains blue in iodine, carbonaceous outer coating, and perithecia, generally globose perithecia, embedded in that stromata, right? And so that ascus apical plug is something worth taking a slight closer look at. Here's some images from an old paper. This is Xylaria nigripes, I believe. And you can see the ascospores, single-celled ascospores, and this highly developed ascus apical plug at the tip of each ascus. And this shape, the shape of it having a kind of a flared outer edge and a second bulge in the middle here, right? The shape is different in different species, in different groups of species. And so you can use this to confirm identities or to arrive at identities. It's a really interesting structure. And you clearly need a microscope to be able to see these things. And it is a shame that you really can't identify Xylarias without microscopy, um, because they're so morphologically variable. And let me point that out for just a minute. You know, when you look at Xylarias, like even a single fruiting, so like all of these are fruiting from the same piece of buried wood. That's why there's dirt on them. The wood was buried. And that each fruiting body is just incredibly different. And that one of the reasons that there are 800 names for Xylarias is because morphological species descriptions were the name of the game until just the last couple of decades, when we've been able to really utilize DNA concepts in speciation, right? And so there's a lot of species that have been described redundantly. Dr. Yuming Zhu, who was a student of Jack Rogers, the guy up in Washington I was telling you about. Yuming Zhu is really, Jack Rogers focused on the whole family Xylariaceae and was really interested in development and evolution, and really he was very interested in the flat ones, in the hypoxylons. Yuming in Taiwan was really interested in Xylaria specifically. And right now he is probably the world expert, like by far the world expert in the genus Xylaria. I spent 3 months in Taiwan with him a couple of years ago, talking about Xylarias and collecting Xylarias and working on Xylarias. And he's great. And he tells me, he says there's 826 names of Xylaria. And he says he's very humble. He's this like little Taiwanese guy who's got this like dry sense of humor. And he says, well, you know, I can tell you about some of them, but there are some species that I can't tell you if they're good species or not, because I have not examined the type specimen. So I can't tell you about 25 of these species because I've never looked at their types. And it took me a minute. I thought, well, if you can't tell me about 25, that's cool. And then I thought, that means Yuming has looked at the type specimen for more than 800 Xylarias. It blows my mind. And so he tells me, having examined all of those types, he thinks about half of them are redescriptions of something else, that they're not good species. So Yuming says there's probably about 400 good species of Xylaria described in the world right now. 400, not 800. So it's still one of the largest groups of ascomycetes, but it tells you something about how much work we need to do to kind of put these in order. And this is in part because there's been, you know, hundreds of people that have been working on Xylaria over the last 2 centuries, and nobody's ever sat down and put it all together and said, okay, what's good, what's not good, what makes sense, what doesn't make sense. Somebody needs to do that at some point soon. I'm working on it. But the point is, in some sense, it's confusing, and one of the most stable characteristics between taxa are microscopic characteristics. This is the microscope I use. This is an old Zeiss WL microscope, and you can go online and you can look at microscopes and you can say, oh, microscopes are just horrendously expensive, which is true. Microscopes are horrendously expensive. This microscope comes from Germany. Zeiss has been making fine optics for a really long time. And this comes from Germany pre-World War II. This is from the 30s. That's where these microscopes come from. And you can, if you spend a little time and a little money, you can assemble some of the best optical instruments from parts. You can get the condenser, the lens system on the bottom, the stage, the nose piece, and the objective lenses. The objective lens is definitely the most expensive part. The headpiece, the eyepieces, the light source, the body of the microscope. If you're patient on eBay, you can put together a microscope like this for under $1,000, which is still really a lot of money, but it's a lot less than you'll spend for a new microscope of similar quality. That's something to think about. Not everybody can do that. Luckily, there are great, generally, there's a lot of new spaces where you can work on things like this. Eugene, where I'm based out of, we've got this thing called 30 Lab, the Thinkubator. I'm starting to work with some. Thinkubator has great lab space. They have microscopes available, and you can use their space by the hour. There are also groups, I know Jay runs Fungi for the People, and right now they don't have a community lab space, but they keep thinking about it. And so I know there are places in the US, I believe Minneapolis, Minnesota, their mycology club has community available microscopes. There are a lot of the Oregon Mycological Association in Portland, Oregon Mycological Society, pardon me, not association, in Portland has a couple of microscopes for use by the group, and they pull them out at meetings. And so there are growing resources for being able to access microscopes and bench space, culture space. The last meeting I went to, again up in Portland, they had a little 1 square foot laminar flow hood for starting cultures from fungi that people had brought in for the meeting, which was really wonderful. And so these resources are growing. So look around your area where you are and see, there are these community lab spaces arising all over the country now, which is really wonderful. And so even if you don't have a microscope, there's potential that there's access to a microscope somewhere near you. And particularly on the East Coast, where there's a higher diversity of Xylaria, it can be a really wonderful thing to look into. And microscopy as a life skill is something I sort of recommend for everybody. It gives you attention to detail, and it gives you this window into the microscopic world that's just kind of mind-altering. It's a really wonderful thing. The other cool thing about Xylaria in terms of identification is that they don't really lose much on drying. And so when you find Xylarias, even if you don't have the ability to examine the spores and make a confident species determination, you can collect them. And in general, in the US, ascomycetes like this, wood decay fungi, because they're not something that is regulated by food foraging, it tends to be pretty unregulated. And the nice thing, like other fungi, the Xylaria fruiting bodies are just the fruit. It doesn't really hurt the fungus at all. The fungus is colonizing the woody substrate, and you can collect the fruiting bodies without damaging the fungus at all. A stout knife helps when you're pulling fungus off of wood. And Xylaria preserve beautifully by air drying. A lot of the fleshy fungi, you really need a dehydrator to be able to preserve reasonably. And they kind of lose a lot of the things that make them characteristic once you've dried them. It's really hard to identify agarics, for example, once they've been dried, unless you're really, really well trained at looking at agarics. Xylarias, on the other hand, preserve almost perfectly by air drying. They shrivel a tiny bit, but you can put them in a paper bag. I know a bunch of people who save all the envelopes from their junk mail to be fungus bags. Put them in a paper bag, let them sit with the bag open. I'm showing you a closed bag, but let them sit with the bag open for about a week or so inside. They'll dry out. Label them with as much information about where they were collected, what they were growing on, what kind of trees were in the habitat, time of year, all of these things. Make a collection, and then you can save it and do the microscopy later when you do have access to a microscope, or you can just put it in a box and you can mail it to somebody who's interested in these fungi. And I personally am pretty much always interested in seeing interesting Xylarias from all around the country. So if you find a Xylaria and you're like, this does not look like Xylaria hypoxylon or Xylaria polymorpha, dry it out, put it in an envelope, send it to me. I will very happily make a slide, take pictures, and pin an identity on it for you, and then mail it back to you. I love Xylarias. Xylarias are great. And so, why should we be collecting and saving these fungi? It comes back to that diversity question again. And so this is, I don't know if you can even really see them on this piece of bark. So this is a piece of spruce bark, and there are tiny little fruiting bodies all over it that I'm holding. This was again from a collecting trip up in Mount Hood in Oregon. And here's a view down through the dissecting microscope of its section. You can see white interior, carbonized surface. You can actually even see these parathecia are underdeveloped, but you can see a couple of mature asci in here. These kind of dark lines are asci with 8 mature ascospores. And so it turns out this little thing, this is actually at the moment classified as a NoMania. But again, we said NoManias are often just flat Xylarias. And you can see these have a really central stalk. This should definitely be reclassified as a Xylaria. But this is, right at the moment, it's called NoMania kellermanii. This is a fungus that was collected in the early 1800s in Ohio and described from that specimen, and has never been seen since until I collected this last year. And so, part of the reason we have all these rare fungi and all these this unknown diversity is that people don't pay attention to these small parathecial ascomycetes very much. But like, who knows how important this fungus is ecologically? What does it serve as a food source for? What role does it play in the decomposition of these trees? This could be an incredibly pivotal fungus to carbon cycling in these northern forests. And we have no idea because nobody's collected it, nobody's worked on it, right? Luckily, in the United States, so Jack Rogers is that guy up in Pullman, Washington, that I told you about. He's extensively, he just retired, but in the 80s in particular, he was very prolific and put out a series of papers on Xylaria in the continental US. And if you look at your mushroom guides, out here I tend to use Mushrooms Demystified. I'm not really sure what your guide of choice is on the East Coast, but probably, I'm going to just make a guess. If you flip to Xylaria, in whatever guide you're looking at, you're going to see Xylaria hypoxylon, Xylaria polymorpha, and maybe Xylaria longipes listed. There's so many more species of Xylaria in the continental US. And so Jack Rogers has been pivotal, why can't I say that word? Pivotal, in the 80s in particular, putting together this series of papers on Xylaria in the US. And so there's a provisional key in Mycotaxon. Mycotaxon, of course, is publicly available through CyberLiber. I'll put a link out with the thing, but you can access this paper for free anywhere in the world and get this key. Ooh, excuse me, my computer is telling me I have things to do. Yeah, you can access this key from anywhere in the world. And this is part of a series of papers that are all cited in this paper, and I've got the citations listed at the end of the presentation. There are papers on each of the major groups of Xylarias, the Xylaria cubensis group, the Xylaria hypoxylon group, the Xylaria polymorpha group. There are different papers dealing with the close allies of each one that are totally worth reading if you're interested in these fungi. Really interesting. And so I just wanted to talk a little bit about the diversity that you get to see in North America. There are a group of Xylarias that grow on woody seed pods, and we have a few in North America. This is a gumball from a gumball tree, Liquidambar styraciflora, and there's a Xylaria that grows on them, host specifically, called Xylaria liquidambar, or Xylaria liquidambaris, depending on who you talk to. The name was published as liquidambar. Somebody thought it should be corrected to ambaris. There's a debate in the literature. I just call it Xylaria liquidambar. Host specific, if you see Xylarias growing on the seed pod of a liquidambar, you know it's this one. Same thing with magnolias. We have this magnolia inhabiting Xylaria, Xylaria magnolii. Really beautiful fungus. Occurs in the southeast of the United States. This is one that has been reported from Texas and Louisiana, the tropical, subtropical species. Florida, you find it in Florida sometimes too. Xylaria tuberoides. It's part of a group of relatively large Xylarias. So these are almost golf ball sized, large Xylarias, along with like Xylaria poetii, which occurs in Florida as well. Xylaria apiculata, and here you can really see the difference between the asexual stromata, where they can be highly branched, high surface area, and the perithecial stromata, where the perithecia are embedded. Xylaria apiculata is named for its little apiculate tips, which you can see here, and they have this really characteristic, the outer coating that starts out covered in conidia ends up kind of cracking and running its lines down the fruiting body. And you can see the perithecia embedded. And here it's swelled up with perithecia, and there's this sterile border. This is a little unusual to have kind of two perithecial zones. And then so this is apiculata. And the apiculata has a relatively short fat spores and a flaring apical apparatus, which helps identify it. This is one from the tropics. There's a whole group of capitate Xylarias, and so I wanted to show you guys some capitate Xylarias. This is Xylaria afcomosa. That there's a great paper from Thomas Lasso in Denmark on this group of fungi, large spored with a wide base to the apical apparatus. And they when they're young, they can have a really interesting yellow pigment, something to watch for if you see them. Physalis, Xylaria physalis, occurs mostly tropical again, but so most of the diversity for Xylarias, I think I said this, but just a reminder, most of the diversity for Xylarias is tropical, and you find higher diversity in hardwood forests than you do coniferous forests. So East Coast, you get a lot more. There's a great couple of papers on Xylaria from the Great Smoky Mountains. So if you live in North Carolina, Tennessee, Virginia, that paper on the Xylaria of the Great Smoky Mountains is great. And there's a huge diversity. There's, dozens of Xylaria that occur in that habitat. Physalis, you find in the US, you find Physalis again, like Louisiana, Mississippi, Texas, Florida. And it's one of the ones that you can relatively confidently identify. If you're in tropical, subtropical systems, you can identify this one confidently based on these horizontal constrictions. Not very many Xylarias have that. So you can see those horizontal constrictions here. And it's a result of how the perithecia develop within. You'll end up with these patches where the perithecia are all kind of in a horizontal line. And then the next a loop of perithecia, and then you'll have a sterile zone. And they're relatively smaller spored with much smaller apical apparati. And so I just wanted to point out there are lots of resources, particularly for North America, but there is no single key. There is no single way to find out what is your Xylaria. You need to dig around a little bit. And so here's that 2008 Pyrenomycetes of the Great Smoky Mountains, number 6. It has the Kretschmaria, the NoMania, the Rosalinia, and the Xylaria. And notice that they've chosen to include Kretschmaria, NoMania, and Rosalinia, which are those 3 genera that arise from within Xylaria, right? And so that's published in Fungal Diversity, which is, I believe, often open access. If you can't get access to this paper, send me an email. I'm happy to send papers, send advice, look at pictures, anything. I love Xylarias. I'm working on a book on cloud forest Xylarias from Ecuador right now, which can get added to this list. But yeah, let me know if you have any questions. And I think that is all I had for talking about Xylarias broadly. So if there's any questions, I'll take questions now. And yeah, thanks for listening. Hey.
Right. I don't know if you can see us or not. Can you hear us at all?
Say it again?
Can you hear us?
I can hear you. It's a little garbled, but I can hear you.
I'm asking if you know anything more about the lignolytic enzymes, or is that just...
Okay. I think we might need to get you closer to the microphone. I heard, do I know more about the something enzymes?
Lignan enzymes.
Oh, the ligniculous enzymes. No, so the lignin decay in Xylaria has been a little controversial. There's a body of literature, mostly coming out of Osono's lab in Japan, showing experimental lignin degradation, where they're inoculating ligniculous substrates with Xylaria, so sterile substrates, no other fungus, and they're showing distinct mass loss of lignin. So they're definitely consuming the lignin. There's a recently published genome for Xylaria hypoxylon, and part of that genomic analysis is that there are no known peroxidase genes in the Xylaria genome. So at the moment, nobody knows what enzymes are doing the lignin decay. That is an area of activity. I know Mark Stadler's lab is working on that, and I know Osono's lab is working on that, but it's unclear at the moment. Nobody really knows.
Great, thank you.
Yeah.
Hey, Roo, I have a question.
Yeah.
Are they strictly white rot, or do they break down both lignin and cellulose?
Both lignin and cellulose, yeah. So the thing about white rot, I mean, white rot fungi in general is that the reason that they're breaking down the lignin is to access the cellulose and the hemicellulose that's bound up with the lignin. And so they're breaking down the lignin so that they can get to the cellulose. The cellulose is like where all the good sugars are, really.
Hello, Roo. Thanks for the talk.
Yeah.
I was wondering about, you said it was cultivated in Japan. Would that be possible from the flesh of a, like white tissue flesh on a petri dish?
Well, so you can't, so culture is one of the nice things about wood decay fungi is they do culture relatively easily. Xylaria is so there's a group in Japan that's done a lot of like experiments with wood decay fungi. Actually, Lynn Bodie's lab in Wales also has done a bunch of interesting competition experiments using Xylaria. I've cultured hundreds of species of Xylaria at this point. Usually, you can culture them from the white interior flesh in a pinch. Usually, ascospores. If you can get a mature fruiting body and break it open so that you can see the perithecia, and then take a sterile needle and scoop out the goop on the inside of the perithecia and plate that out, you'll get really lovely cultures. They culture nicely from the ascospores. The other thing, there are groups, there's Sarah Robinson's lab at Oregon State University, works on wood spalting. They're like a pigment lab, and they're interested in Xylarias because Xylarias are spalting fungi. And so they've been culturing Xylarias directly from decaying wood. So they're taking chunks of dead wood and just culturing the fungi out and isolating the fungi. And so you can get the Xylaria to grow from just about anything where the mycelia is. They're very active little fungi. Does that help?
Growth of the is the mycelium? Is it ropey? Is it cottony?
You get sometimes they don't really make the kind of like rhizomorphic ropes that you get from some of these wood rot basidiomycetes. In culture, you tend to get relatively non-dendritic, dense hyphal fans. It's very white and very silky, not terribly much aerial tissue. and they will form stromata primordia. They'll form the initials in culture really easily, depending on the species. So like Xylaria hypoxylon, if you wanted to grow Xylaria hypoxylon on a petri dish, I just use 2% malt. It grows great on 2% malt. And they'll send up the little conidial fruiting bodies, but they don't produce perithecia in culture. They're all sterile fruiting bodies. And so I feel like I've got some pictures of Xylaria in culture here somewhere. Let's see. Hold on just a sec. I'll see if I can find one. That's not it. Well, might be on my other computer. Oh, wait. I got one more idea.
Can I ask another question?
Yeah.
Do you know about the Pororia species?
Poria? Yeah.
Do they always have caps on top of like those antlers?
So it's like Poria and Podosordaria are, yeah. So here, there's a great resource, the home of the Xylariaceae, which is run through Dr. Yuming Zhu's lab in Taiwan, has some information on these things, and they are, where are they? Accepted genera. They're caps in the same way that Xylaria comosa with the capitate head. So it's still embedded perithecia within a stromata, but they're, yeah. Okay, let's see. Let me screen share this. Screen share. And then share this. So yeah. So here, it's mycology.seneca.edu.taiwan/Xylaria. We'll get you there. And there's information about a bunch of these things. And so the Poronias grow on dung, and this cap, and it's all dried out. It's actually rounder when they're wet. But you can see the osteoles for the perithecia, which is much like the Xylaria. It ends up being perithecia embedded in a stromata. Does that make sense?
Yeah.
Cool. Yeah.
Magnolia species, and I was wondering if you could talk about why they're growing on the flower structure.
Wait, so that was a little bit garbled. Say that again?
Firstly, what was the name of the magnolia inhabiting Xylaria? And then why does it fruit on the flowers?
Oh, I see. So the Xylaria, the seed inhabiting Xylaria are really interesting. They grow on these seed pods, and there are a couple of I don't know. I think it's in that list of further reading. And I'll make that presentation as a Google document that I'll just make public. There's these seed pod inhabiting ones are host specific because the substrates are relatively specific. And Dr. Rogers up in Washington has been working with Xylaria carpophila, which fruits on the beech mast, the little beech nut husks. And he has, I mean, you notice when you find these things on the magnolias, on the liquid ambar, on the beach mass, or there's one in the tropics called Xylaria xanthinovillutina that fruits on big legume pods in these fabaceous trees. And when you find them everywhere. You find them on like almost every downed pod. And Jack thinks that what's going on is that they fruit out in the spring, and they're that conidial, white powdery coating in the spring when the tree is in flower, and the conidia get into the flowers and germinate and grow down into the developing fruit, and then wait until the fruit is fully developed and falls, and then the fungus is already there and able to utilize that substrate and grow out again. And so it's this cycle where the conidia serve as an infective propagule that gets in the same way that pollen gets in through the flowers. And so it's this kind of deep evolutionary, co-evolutionary story that's still it's Jack's theory. As far as I know, it's not been published yet, that theory. But it sounds like a good one to me.
Very
Does that help?
Yeah.
Hey, Roo.
Hi.
Hey, I was just wondering, you mentioned that the Xylaria genera is like one of the most diverse. What are the kind of like key defining characteristics to ID something as Xylaria?
Okay, so to ID something as a Xylaria, you're looking for upright stromata at the moment, unless somebody decides that they want to lump NoMania in there. But upright stromata, or I guess maybe actually we should start a little further back. Stromatic ascomycetes, so parathecia embedded in fungal tissue. Multi-parathecia, you pretty much always have more than one parathecium. Carbonized outer tissue, so they're going to be kind of crunchy with a woody texture. Crunchy on the outside, woody texture on the inside. 8 dark spored ascospores. And then what else holds it together? Yeah, I think those are your major defining characteristics. Upright stromatic ascomycete, carbonized, single-celled ascospores, melanized, ellipsoid and equilateral.
Okay, thank you.
Yeah.
Where are you?
Eugene, Oregon.
Cool. Can we get your email to contact you about some of those resources?
Oh, absolutely. Here, let's see. I will, let's see. What's the best way to do that? Well, let me hear. I can probably kill 2 birds with 1 stone. Actually, let me give you my Gmail address. Yeah. So, do this. So I'm going to throw my email address up on the screen share. Oops, wait a minute. That didn't work, did it? Doing this wrong. Sorry, guys. There it is. So, so this is what Xylaria looks like in culture, incidentally. But there's my email address. I'm also gonna. It'll be on the on that slide. I'm going to make that Google presentation that I just gave public, and give the link to Jay, or to Peter. So hopefully that can get distributed, and then everybody can have access to that presentation that wants it. There's my email address. Again, feel free to contact me about anything Xylaria, Xylariaceae. Totally happy to help. All right. Everybody get it?
You know what you're talking about. Yeah.
Or like the giraffes.
No deals, no forms. It looks like one of those like wasps or dolls. Yeah, so it's kind of like all around.